I am Nathan Lewis. I am a professor at UC San Diego, and in my lab we do a lot of computational modeling of all the processes that are involved in cell growth and interactions of the cell with its surroundings. So one of the things that excites me the most in this space is basically we're able to have an understanding of how living organisms work at the molecular level. And then beyond that, we can, after knowing how these things are wired in a way, we can work with them to be able to improve, to rectify problems such as you see in disease, or even engineer certain cells that are used for producing high-value pharmaceuticals. There are a lot of challenges and opportunities. Our understanding of all of these pathways is still very rudimentary. We have decades of biochemists that have characterized the specific pathways, which enzymes are contributing to them, and a number of the physical constraints on their activities. However, there's still a large portion of metabolism that is inadequately or incompletely characterized. And so we really need to have a lot of work on the biochemistry to further develop this. But again, a lot of this work has been done one enzyme at a time, and so we need to develop novel approaches to connect all of this information. And that's where these genome-scale metabolic network models are quite valuable, and a lot of the algorithms that people have developed to use them is that it takes all this information and brings it together. The models themselves still take into account predominantly metabolism itself. And so what we're missing are all the connections of metabolism to gene regulation. We're missing the connections of the alterations of metabolism to protein synthesis. We're missing a lot of to signaling pathways and to many other processes, even to the point of we're missing interactions with metabolites and how they modulate how one cell interacts with another. And so these are all great opportunities to experimentally determine all these, but also to develop further computational frameworks to analyze and simulate those interactions. In our work, we find some of the most useful data starts with understanding how metabolism is changing outside of the cell. Because once you understand how it's changing in and outside of the cell, that allows you to then interpret and predict what the cell is actually using and what it's secreting. So that gives us like a starting point. Then beyond that, we like to use all sorts of different omics data internally to understand which pathways are active and so forth. And furthermore, we like to look at that intracellular data to understand how changes in pathway and the changes in metabolism we may be predicting with our algorithms are ultimately changing the gene regulatory programs of the cell. In our group, we developed a lot of models in particular for working with cells that produce a lot of pharmaceuticals. So if you look at the top 10 drugs by cells globally, many of them are proteins that are actually produced in mammalian cells. And so for us, as far as looking at metabolism and translating to the patient, comes down to using these models to figure out how these mammalian cells are using the metabolism to produce the drugs that ultimately will go to the patient. So it ends up becoming a great opportunity in it because that's a system that you can actually engineer to drive down costs and improve quality of the therapies. A lot of diseases are complex in that there are many genes involved, many factors involved, and we, with our minds, cannot fathom that many or process that much information all at once. And so now we have the tools, though, where we can at least build the models, use those to interpret data, and allow us to then come out with actionable discoveries and treatments.